Hi, we're from Lab 13 Chester, and today we're going to show you how to do your own Lab 13 DNA extraction. You will need some salty water dyed with blue food colouring, freezing cold nail polish remover, some washing up liquid, a cup for each person you're doing it with, and 2 test tubes. Right. First, these are my volunteers, Connor and Freya, and I'm Lara. So first, they're going to have to scratch at their cheeks with using their teeth, so that they can get their cheek cells off of their cheeks, so we can see the DNA. Now, what they're doing is they're breaking away the membrane, and there's 3 layers to a cell. There's the membrane, then there's the nucleus, and then there's the DNA right at the centre. So they're just scratching away the membrane right now. Right. Now what I'm going to do, I'm going to pour a little bit of the salt water into the cups. Now that they've got all the cheek cells and they're swelling around in their saliva in their mouth, they're going to have to swish this salt water around in the mouth to collect all the cells for the test tubes. Now there will be quite a bit of dribbling, that's why you will need some tissues on standby. Right. 3, 2, 1, go. As you can see, it isn't of the nicest, but we have to do it.
I smacked most of it on the tissue. Ugh.
Right. Freya's just going to have to do a little bit more because we need it about up to there.
Okay.
But we'll leave it for now. Right.
Ugh.
Connor?
I don't want to do that again.
Right. Now in there should be loads of little cells broken up down to the nucleuses. Right. Now what we're going to do, we're going to get this washing up liquid and the pipette. Now we're not going to use the pipette normally. We're going to just dip it in, push it around a bit, and place it in to the test tube and slowly just mix that around. You don't want it, try not to make any bubbles if you can help it, because we don't really need those. Right, now you have to get your freezing cold nail polish remover and use the pipette normally, so squeeze the bubble and not suck some of it up. Now you've got to tilt it on its side so that it drips in very slowly, because if you do it too fast it will mix, and we just need it to form a layer on top. Your layer will need to be at least a centimetre or 2 thick. So, but try not to mix it. As you can see there, now it's got a see-through layer and a blue layer. Just going to do this with Connor's. Now what we're just going to do, we're just going to move the test tube around like you're drawing an 8, very slowly. Now if you look carefully, there is a strand, little silvery piece of string going from the cloudy layer up to the top of the clear layer. The little bubbles on it aren't your DNA, but the silver strand is Freya's. And that Qantas has come up really well. You can see it just on that side. I hope you enjoyed our experiment and I hope you try it. Remember, you might have to have an adult to supervise you, but it's a really fun project to do. Thank you for watching.